This video is going to go over the process of meiosis. So meiosis is a different type of cell division than mitosis. In meiosis, it's a type of cell division that's going to eventually result in 4 genetically unique haploid daughter cells. So there are a couple key things here. The first one is that they're going to be genetically unique. And the second one is that they're going to be haploid. And haploid means that they contain half the number of chromosomes. So a normal human somatic cell has 46 chromosomes. So after meiosis is over, the haploid daughter cell will only have 23 chromosomes. Meiosis only occurs in sexually reproducing organism, and it is the process of making gametes. And for humans, those would be egg or sperm cells. So those contain half the amount of genetic information. 23 chromosomes in the egg, 23 chromosomes in the sperm. They come together and they'll create an organism that has 46 chromosomes. Now, this 23 and 23 is pretty important because we get 2 copies of every trait. So we get 2 copies of all of our information, and we're going to talk a little bit about why that's important when we start talking about homologous pairs. So before we get there, I do want to mention that those genetically unique haploid cells, meiosis allows for more genetic variation in its daughter cells due to a number of things. The first one is mutations. So mutations can happen anytime you replicate DNA. So it happens during mitosis, so it also happens during meiosis. But then meiosis also has 2 additional ways in which genetic variation comes about. The first one is crossing over, which happens during prophase 1, and I'll talk about that when we look at the picture. And the other one is independent assortment. And so in crossing over, what happens is homologous chromosomes will exchange segments of DNA. In independent assortment, the chromosomes are passed on independently of one another. So I'll kind of show what this looks like once we get to those stages. So in meiosis, you start with a normal parent cell. In our case, we're going to pretend that our organism only has 4 chromosomes. So in our organism, if it only has 4 chromosomes, then we're going to say that it has 2 homologous pairs of chromosomes. So we're going to say that in this particular instance, that this is chromosome number 1, and this one came from its mom. And then it has another chromosome number 1, but this one came from its dad. And then it has a chromosome number 2, and this one came from the mom, and it has a second chromosome number 2, and that one came from its dad. So it's sort of like us. We have 23 pairs of chromosomes, so 46 altogether. We have 2 chromosome number 1s. One came from mom and one came from dad. So we're going to pretend that this is how this organism works as well. 4 chromosomes, technically 2 pairs. 1 pair from mom, 1 pair from dad. Okay, so the first stage of meiosis is very similar to mitosis. The very first thing we have to do is replicate DNA. Now, this DNA replication will only happen one time, and if you remember way back from the notes, when chromosomes replicate, and I have it written down here, remember this is 1 chromosome with 1 chromatid that contains all of the information. When DNA replicates, there will be a second chromatid. This is still called a chromosome. All right, so over here we can see that both of our chromosomes, all 4 of our chromosomes, have replicated. And in this case, in prophase 1, what happens is our homologous pairs pair up. So that's a little bit different than in mitosis. So what we see here is homologous pairs of chromosomes pair up. So this was chromosome number 1, remember? But we've got moms and we've got dads. And this is chromosome number 2s. We get 2 of them. Again, 1 from mom and 1 from dad. All right. So what ends up happening here is we get a little bit of crossing over. And so this was in the notes about how we get some genetic variation. And so chromosome number 1 carries a set of information, and the information you get from your mom is the same as you get the information from your dad, but maybe a slightly different variation. So let's say chromosome number 1 codes for eye color, freckles, and hair color. Those 3 genes are on this chromosome as well as this chromosome, but you might get different versions of it. Your mom might give you blue eyes, your dad might give you green eyes. So the interesting thing that happens during this step is that because they are so close together here, and they have all this overlapping, segments of DNA will break off and exchange. So we get little bits of information that break and exchange from our pairs of homologous chromosomes. So that happens to this pair of homologous chromosomes. That's also going to happen to this pair, and it can happen anywhere, and it's different amounts, and it just sort of depends on that particular cell at that particular moment. So we've got crossing over events that happened here and crossing over events that happened here, so there's that exchange of information. So that's one of the ways in which our gametes at the end are going to end up being very unique from one another. Now, once our homologous chromosomes have sort of paired up, what ends up happening is our homologous chromosomes end up pairing up at the metaphase plate. So homologous chromosomes, homologous pairs, line up at that metaphase plate. And this is called metaphase 1. And we have little microtubules that help them get there, but notice that they're in pairs, so this looks very different than mitosis. The next step is that our homologous pairs get pulled apart. So homologous pairs get pulled to opposite ends. And then after that happens, we've got telophase where we get a little bit of cytokinesis, so we get that pinching of the cell membrane, and we basically form 2 cells here. Now, these cells are already considered haploid, because technically there are only 2 chromosomes in them. This is one chromosome, this is another, this is one chromosome, this is the second. But remember in this case that these chromosomes have 2 matching chromatids on them, or somewhat matching. So we've got more information in these cells than we need. So what ends up happening is that now our chromatids need to split apart from each other. So then we have metaphase 2. So we have our second set of division that's going to happen. So in metaphase 2, our chromosomes that are left are going to line up at the metaphase plate. And keep in mind, this is happening in 2 different cells because I've already had 1 cell division. So now that my chromosomes have lined up on the metaphase plate, now during anaphase, my chromatids are going to split apart. Okay, and so that's anaphase 2. Telophase, we get that cytokinesis that happens again, so our cells start to pinch. And what you'll notice is that these are still haploid, right? They still each have 2 chromosomes, just composed of 1 chromatid. And then once cytokinesis is finished, we'll have 4 new cells at the bottom. They are all genetically unique and they're haploid. So they all contain 2 chromosomes, but they're all different from each other. So if we look at just these 2, right? So here is a chromosome, and due to crossing over, it has this set of genes down here. And then here's its second chromosome. So chromosome number 1, chromosome number 2. This chromosome number 1 and this chromosome number 2 look different. If we keep looking at our gametes, this chromosome number 1 and this chromosome number 2 look different than the other cells. And then here's my last one. Here's chromosome number 1. Here's chromosome number 2. That looks different than even the other cells. And this variation is really important because the fact that we can make so many different versions of eggs and sperm is the reason that your siblings look similar to you, but not the same. So you get all your genetic information from your mom and dad, but you get all of these different variations of their information passed on to you. So that is the process of meiosis. Before we finish, I just want to mention that independent assortment again. So here I've redrawn anaphase 1. And telophase. Anaphase 1 and metaphase 1. So I'll just label those on here. And it's during this time that we see the chromosomes start to assort themselves. So you'll just notice that if we're paying attention to what's happening, that these chromosomes right here are going to end up together. And then these chromosomes over here are going to end up together. So we have this like mostly red one and this mostly purple one that are going to end up together, and then those are going to end up making gametes. But then we have this mostly orange one and mostly blue one, and then they're going to end up making gametes. But this is not how they have to arrange themselves. So if we pay attention just to these bottom two, because those are the only ones I moved, if they flip-flop, if they arrange themselves differently or independently of these two, so these arrange themselves however they want, what's going to end up happening is that these two instead are going to get passed along with each other, whereas these two will get passed on along with each other. So this time the red and the blue get passed on with each other. See the red and the blue here. Whereas in the other gametes, it's the orange and the purple that get passed on with each other. And so these look different, right? So those are going to end up making different looking gametes. And that's because the chromosome pair number 1 and chromosome pair number 2 can assort themselves independently of each other. So it doesn't matter which chromosome number 1 you can get either of those chromosome number 2s depending on how they assort themselves at that metaphase plate. So we'll talk more about this during class, but I just wanted to give you a brief set of notes on how meiosis works.